Like to begin the session with the quote. I quote, "Almost everyone are enthusiastic about the promise of biotechnology to cure the disease and to relieve sufferings." I unquote, says Leon Cass. A warm greetings to you all for the webinar organized by Department of Biotechnology, Sri Bonendra College, Karkala. Myself, Mrs. Deepti, Head of Department of Biotechnology, would like to commence the webinar by heartily welcoming everyone on this platform. A warm welcome to you all for one-day national webinar on high throughput screening for drug discovery. It's my great pleasure to welcome today's speaker, Dr. Krishna Kurtukoti, Faculty Fellow, Mycobacterium Research Laboratory, Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram. Welcome you, sir.
Welcome you, sir.
I would like to welcome the chairperson of webinar, our beloved principal, Dr. Manjunath A. Kotian. I also would like to extend a cheerful welcome to our dear participants without whom our event wouldn't be complete. Welcome you all. Now I take this privilege to introduce today's speaker, Dr. Krishna Kurtukoti, Faculty Fellow, Mycobacterium Research Laboratory, Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram. Dr. Krishna Kurtukoti completed his PhD from Indian Institute of Science in 2010. His doctoral research was focused on understanding the importance of DNA repair in Mycobacterium during stress condition. After PhD, he carried out postdoctoral research at Sanford Burnham Medical Research Institution, San Diego, California. The topic: investigating the role of small RNAs in gene silencing and modulating the innate immune response of Drosophila melanogaster. Subsequently, he undertook another postdoctoral position at Rutgers University, New Jersey, on the topic studying iron homeostasis in the human pathogen Mycobacterium tuberculosis. Later, he was awarded the Ramalingaswamy Fellowship from Department of Biotechnology, which led him to initiate his independent research as a faculty fellow in Mycobacterium Research Laboratory at Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram. His current research is aimed at investigating evolution of drug resistance in bacteria, understanding the mechanism of drug resistance in bacteria, and understanding the mechanism of iron homeostasis. We welcome you again to this auspicious event, sir. So before commencing our session, I request our dear principal to share his keynote address. Please, sir.
Good morning, ladies and gentlemen. Honorable resource person of this webinar, Dr. Krishna Kurtukoti, convener of this webinar, Madam Deepti, Head of the Department of Biotechnology, and dear participants. At the outset, I welcome you all to this national level webinar on a very novel topic. Ladies and gentlemen, as we all very well aware, drugs play a crucial role in treating any health problems. Accordingly, finding out the most suitable drug for a disease is equally important. And it also involves untiring efforts of all. In order to find a very effective drug, n number of experiments and trials need to be done. Only then a suitable drug can be released to the market so as to treat that particular disease. Absence of drug these days for COVID-19 has made all of us to understand the importance of drug discovery. Our wait for drugs has almost made all desperate and disheartened. At any cost, we want to put an end to this horrible situation. Today's webinar topic rightly emphasizes the importance of drug discovery and will help to understand screening process that is necessary in drug discovery and saving numerous lives. Friends, technology in general and biotechnology in particular is used in fastening drug discovery process. I have heard that HTS, high throughput screening, commonly used by pharmaceutical and biotechnological company to identify compounds called hits with pharmacological or biological activity. It allows quick and efficient screening of large compound libraries at a rate of few thousand compounds per day. This helps in increase of efficacy and reducing of time involved in discovery of drugs. Hence, this webinar is special to understand this HTS process. Ladies and gentlemen, we have a very learned scholar of very reputed institute, Dr. Krishna Kurtukoti, to enlighten us on this topic. At the outset, I welcome you, sir, most respectfully, Dr. Krishna Kurtukoti. I also welcome all the participants, welcoming one and all. I welcome my friends in the teaching fraternity, welcoming one and all. Over to Dr. Krishna Kurtukoti. Sir, please.
You are in mute, sir.
Yeah, yeah. Hello, am I audible?
Yes,
Okay.
sir. Yes, sir.
Yeah. First of all, thanks to Professor Manjunatha for giving me this opportunity to present this webinar. Let me share the screen. The screen sharing has been disabled. Can we activate that?
Yes, sir. Fine. Yes, sir.
Okay. Okay, so the slides are visible, right?
Yes, sir.
Okay, great. So yeah, first of all, thanks Professor Manjunatha for giving me this opportunity to present this webinar, and a special note of thanks to the HOD, Dr. Deepthi and Pratiksha for coordinating this event. And these are actually really tough testing times. And so I hope all the participants are safe and doing well wherever they are. So with this, the topic of today's discussion is high throughput screening for drug discovery. So before I begin, I just want to just emphasize, this is not specifically, the talk is not aimed at the techniques used for drug screening against COVID or something. So it's kind of a general overview as to how, if someone wants to do a drug screening, a drug discovery through high throughput screening, so what are the methods that are involved? What are the things that one should take care? So it's kind of a general introduction to this field. Okay. So before I begin, this is the place where I work, Rajiv Gandhi Center for Biotechnology. And we are currently still a small group with two students, Shalini and Sinchana. And we work under the Mycobacterial Research Group umbrella. So the research from our lab is supported by various funding agencies from India. And as I mentioned, this is a topic outline. So the idea is we spend some time getting introduced to the principle of high throughput screening. Then we look at library of compounds, which is the key part of the high throughput screening. We'll also spend some time understanding the assays that are used for high throughput screening. And then we'll spend some time in how once you do your screening and you get a target or a lead molecule, how do you identify the target? So which process is it targeting? So how do we understand finding those? So without much ado, so screening as such, the term screening is formed a pivotal part in the field of genetics and the modern biology in general. So I think most of you are aware of Mendel's work, who is regarded as father of genetics, and the success of his work, wherein he used pea plants for his experiments, was due to his careful selection of different pea lines or pea strains, which we can call, that had distinguishable traits. So it could be like a round seed morphology compared to wrinkled, or say a pod color of yellow or green and so on. So he screened for these lines and then did his experiments, and that was a key part for the success of his experiments. So subsequently, Thomas Morgan, who worked on Drosophila, and he actually rediscovered Mendel's genetic principles, and he worked on Drosophila, and even his success story was due to selecting powerful mutant lines in Drosophila. So screening as such has been a very important player. The concept of screening was very important in the modern biology and genetics as such. So all throughout I was talking about an observable trait, distinguishable character. So what is the molecular mechanisms for this? What is the molecular reason underlying the observable trait? So is my audio, it's fine? We are good? Okay.
Yes, sir, fine.
Yeah. So this is a schematic which probably many of the students and many of the faculties would know, which is called central dogma of molecular biology. And so what you have here is a genetic material, that is DNA, which gets transcribed into mRNA, then translated into protein. The protein is the actual effector molecule which performs a cellular function, giving rise to the observable trait, which we call as phenotype. So you can see that there are two terms here, genotype and phenotype. Genotype refers to the essentially the genome sequence or the DNA information, and the phenotype is a ensuing observable trait due to the gene expression. So let us look at an example as to how genotype governs a phenotype. So this is again the fly model. So there is a phenotype called eye color. So essentially the phenotype is a trait which calls the eye color. Under wild type flies have a pigmented eye, which is kind of red. And if you have a mutation in the gene that encodes this eye color, essentially what would happen is there is a defect in the transcript which is translated into a defective protein, and as a result you get a colorless or a white eye phenotype. So this is mutant. So this is how the changes in the genotype governs or affects the phenotype. So today's topic is about use of molecules, small molecules in therapy. And this field began in early 1900s by this important work from Paul Ehrlich. He's a German physician, and he thought of this concept of magic bullet. So the idea of magic bullet is you use a small molecule, mainly chemical molecule, which will specifically target an infectious organism without affecting the host. So his work led to the discovery or synthesis of an arsenic-based chemical molecule, which was used to cure or to treat syphilis. And for his contributions in the field of chemical biology, in the field of chemotherapy and immunology, he was awarded with the Nobel Prize. So subsequently, in the early 1990s, Stuart Schreiber, who began as a chemist, collaborated with his other co-workers, and began with an interdisciplinary field which we call as chemical biology. So essentially, the concept here is use the concepts of chemistry to address biological problems such as signal transduction or other cellular process. So this is a very recent development. The field of chemical biology is around 25 years old and has a lot of importance as we go ahead. Okay, so again, what is the principle of this concept in chemical genomics? So we already saw this schematic, that is the central dogma, that you have a DNA, it gets transcribed into RNA and gives rise to protein. So if we have a disease condition caused due to a defective protein, so the idea of chemical genomics is to employ chemical small molecules which can affect the phenotype and then give a kind of can it be used as a therapy or a cure, so on. So it is the use of small molecules to intervene in the gene expression process. Okay, so now what is the relevance of this chemical screening or chemical genomics? So I think the entire audience would definitely appreciate the current situation of the pandemic, where the entire global pharma and academic community is striving hard to identify molecules or therapeutics against the virus. Of course, this is not just limited to the COVID situation. You have in molecules or drug molecules that are designed against several infectious agents. And towards the left, you also have a lot of genetic diseases in humans, which cannot be changed, right? Because you cannot tamper with the host DNA or the genomic DNA level, but you can always come up with molecules that can mitigate the effects of the disease. Okay, so these are this slide shows a list of chemical molecules or the drug molecules which were introduced by several drug companies, and these were used against cancer, HIV, and several other human diseases. And what I would like to bring to your attention is to these two columns. So you see on the top there is a year when the HTS or the high throughput screen was run, and a year when it was approved for human use. So you can have a kind of look that there is almost 5 to 10 years of gap from the time of high throughputs being initiated to the time of approval. So it's a pretty long duration. So why does it take so long, right? So if you have a chemical, why can't you just introduce to the community and cure the disease? So it all starts with the basic research, wherein people like us perform experiments in the lab, and we identify a gene or a pathway which has the potential of being targeted for drug therapy. So this involves much of the basic research work, and then you will identify a drug target. So now once you have identified a gene or a protein pathway, whatever it is, you perform high throughput screening. And at the end of high throughput screening, you get initially what is called the hit. So as Professor Manjunatha said, that hits are what initiate the next line of questioning. So once you have a hit, which is essentially a chemical molecule, we try to see if it has a biological activity as such, because hits that do not have biological activity are not of our interest, and hits that have a biological activity, now we call them as lead. So the lead molecule is not the final drug molecule that ends up in the market, but it has the potential to target to be used as a drug. So then the next step involves optimization of the lead. So you make it more potent, less toxic, or probably, highly permeable, and which can be better than its parent compound. So once the lead is optimized, then you go for the clinical trial. I think you must be hearing a lot of news about clinical trials of the vaccine, and you might be aware that the regulatory agencies are kind of relaxing some of the clinical trials so that the vaccines or drugs end up in the market soon. But technically, a clinical trial would take quite a bit of time before approval. So now, having understood the process of drug discovery, high throughput screening is a widely used method to screen potential compounds to identify drugs. So the idea is, why do we want to do high throughput screening? Why can't we just take some molecules, synthesize in the lab, and just use them, right? So it seems it's predicted that there is a huge chemical space. So in the sense that there is around 10 raised to 60 possible chemical molecules that can be generated. And out of this huge number of molecules, we have currently around 10 raised to 7, probably million to few million compounds that are currently available. So high throughput screening enables us to test this huge number of molecules in a short span of time and identify or come up with compounds which have very important bioactivity. Say they could be antibiotics, inhibitors of signal transduction, anti-cancer, pharmacological substances, and whatnot. So that is the power of high throughput screening. You can analyze, run a screening program with millions of compounds, come up with very important ones from a million or so. So with this small introduction, we now go to an important aspect of the high throughput screening, which is the library of compound. Okay, so what is library of compound? The name as well suggests that it's a collection of compounds. The collection of compounds, the number can vary from few thousands to several million. And so the library of compounds can be further differentiated or separated, classified into a discovery library. So the discovery library is chemically and structurally diverse group of molecules. So the idea behind this discovery library is to test a different family of compounds so that you can work out whether there is a lead molecule that can be obtained from it. So now once you do this, use this discovery library and get a kind of hit, then you use what is called an optimization library. So we saw earlier in the schematic that there's an optimization step. So here the optimization is essentially to improvise upon the lead molecule to make it say more active, less toxic, and so on. And usually this optimization library consists of chemical molecules that are very close to the parent molecule. So essentially it will have some side group changes or say some addition of a functional group and so on. So which enhances the potency of the lead molecule. So what is the composition of this library? What is it made of? So it is usually made of chemically synthesized molecules. It can also incorporate natural products like secondary metabolites. And most of them, the molecules that are used in these screenings are kind of having synthetic drug-like molecules. So they could be having some properties of drug-like molecules and they are used in the library. So an important aspect of the library of compound is that these molecules should be of high purity, because when you're screening, you don't want a mix of compound and then you end up with an ambiguity as to what is the actual compound effective principle. So essentially, when you are performing high throughput screening, like a leaf extract or some plant extract won't work out because they always come up with a bunch of chemical molecules. So even if you're planning to do a screen through that, you are required to separate them, purify each of the components, and then go ahead to test them. Okay, so there is little bit of there's a very little involvement of a regular biologist in the compound synthesis, because this is the domain of a chemist, and this is where the chemistry part comes in chemical biology. So it is usually the organic and the medical chemist who come up with the idea behind what kind of compounds need to be synthesized, and they are the ones who have the technical know-how as to how to synthesize the compounds and so on. And so in order to generate these compounds, we are kind of talking in the range of millions of, or hundreds and thousands of compounds. So it is very difficult to synthesize in the traditional way. So there are some popular methods employed, which we will go through in a little bit of detail. So the popular methods are the solid phase synthesis and the liquid phase synthesis. So these are generally used for generating the library of compounds. So let's look at the solid phase synthesis. So this was originally developed to synthesize peptide-based molecules. So this is a kind of schematic representation as to how this process is done. So you have a solid surface, which is depicted by this gray molecule. So it is essentially a solid support to which you attach your reactant or the monomer. Okay, so the monomer has two reactive groups. So one group will attach to this solid support while the other one is protected. So you don't allow that to be free to get reacted. So the first step is to attach this blue reactant molecule to the solid surface. And once the reaction is completed, you deprotect this protected group. So essentially you can allow the reaction to proceed. So once you have deprotected, you add the second ligand or linker or next monomer, and you do this reaction. So as a result, you will increase the length of this parent molecule by 1. And you can go on doing this repeatedly, and you can eventually attain the molecule of interest by extension. You can also use different mix and matches. So like you have one solid support, you can also take another solid support, attach another molecule, and you have molecule A and molecule B, and mix them together, so that way you can increase your diversity of compound. So there is a lot of flexibility in this method. You can also use, instead of single monomer reaction, you can do you use 2-mers or a dimer. So there are different options available in this method. So let's look at what is the advantages of this method. As I mentioned, there is an option that each bead can be conjugated with different reactant molecule. So the idea in this small molecule screen or generating the library is to come up with a diverse group of molecules. You don't want a same family of molecules. So it's always good to have a diverse group of molecules. So since you have the option of taking different reactants and mixing in the same vessel, you can expect a lot of diversity. So that's an advantage. And the other thing is, since your reactants, the reaction products are bound to the solid surface, it is very easy to purify. So you can just put at the end of your reaction, you separate out your beads, then you cleave the linker, and you have the product of your interest. And another important aspect of this is it is feasible to automate. So essentially, you don't have to sit and do this reaction. You can kind of come up with programs so that will enable to do this reaction. So there's a possibility for automation. So these are some of the advantages of solid phase synthesis. So we next look at another popular method, which is the liquid phase synthesis. So as the name suggests, so you don't have any solid support here. So everything is happening in a liquid interface. So you have a scaffold molecule, which is in a liquid or a medium, which is conducive for the reaction. So then you introduce a ligand molecule with a protected group. So this is called a PG, is your protected group, right? So now you have the other reactant reactant group that is activated, and you perform a coupling reaction, or you just allow this linker to go and attack, and then you extend it. So you drive the reaction to completion, and any left out reactants are flushed out by a kind of a molecular sieve. So any left out reactants are flushed out. So once you have done this, you then release this protected group by chemical processes, and then you kind of generate this hydroxyl group. So that is essentially showing that you have deprotected it. So now once you have deprotected, you then add your next ligand, right? So then you do the same reaction. I mean, you continue it. Then you will have got from n+1, you have got to n+2 and n+3 and so on and so forth. You can keep on extending. So another advantage, a major advantage of this liquid phase of synthesis is that since everything is happening in the liquid interface, it is you can just do in parallel an HPLC based monitoring. So you can, as well as the molecules are being synthesized, you know how pure the compound is, and so that way you don't have to purify and isolate and recheck the purity. So at each and every step, you know the purity of the compound. So you can either tag on to HPLC or other advanced method, put it on to the pipeline of a mass spec, and you can continuously monitor compound purity. So now, whether you choose a solid phase synthesis, a liquid phase synthesis, or whatever was your interest of synthesis, you end up with a library of compound. So now let us assume you started with scratch, you took one of the approaches, and you synthesized a library of compound. So what next? What is the next step? So now you have made a library of compound. So this is a schematic as to how it happens in an industry or in a high throughput management of the compound. So first and foremost, you will try to figure out how pure the compound is and what is the yield. So these are kind of regular chemical chemically processes. So you find out the purity of the compound, you find out how much you have got the yield, then you will kind of evaporate. And since you're dealing with thousands of compound, you have to catalog them in a effective manner so that you can always go back to the compound once you have done the screen. So there should be some effective methods of cataloging, and then they are stored under controlled condition so that you can have a good shelf life. And again, you have to monitor the shelf life, no doubt, because you don't want to use a library which has already gone bad past the expiry date, right? So then you cannot, your results are not trustworthy. So now this is kind of a picture, overall picture as to how what is the magnitude of the compound library. So this is a old data dating around 2009, but you can still appreciate the kind of library of compounds the pharma industries had. So you have some of the big players, Pfizer, Novartis, GSK, and whatnot. So you see that they started out with around 500,000 molecules, and just by around 2009, it was close to 2 million molecules. And now it's almost 10 years now, so you can expect they should be close to around 10 million molecules or so. So we must understand most of this are pharma driven um, projects, and so many times the molecules are not freely available to academic institutions. So you cannot just go and get, of course, many a times they may end up having a collaborative work where they may allow us to get access to this compound, but most often the corporate and research are kind of bit separated out. So having understood the power of high throughput screening, many academic players have entered this high throughput competition. So I'll just share a few examples. So one of them is the Broad Institute. So this is in the Boston area, part of the Harvard University cluster. So they have around 800,000 plus. And they not only give you this compound, they also help in performing a high throughput screening by state of art of the facility, which involves automation and use of robotics. And they also help us in identifying the lead, and if you require target identification. So there is a lot of involvement of this Broad Institute with an academic project. So it's used, of course, it also encourages um, um, corporate collaborations as well. So there's another place called Scripps Institute. This is in Florida. So both of these are from US. And so this Scripps Institute also provides assistance in performing high throughput screening, and they have around 600,000, 700,000 plus compounds. And you see that when you go for high throughput screening, you are no more doing regular pipetting, one pipette and one well per minute. So you're using robotics, so you do a lot of reactions at the same time. So usually this is done at the robotics level. Okay, so now let us say you are not you cannot do a high throughput screening of the magnitude that we discussed, but nonetheless you are interested to screen for some of the projects that you are doing with. So there are commercial vendors who can supply you with molecules. So one of them is the Merck, or formerly Sigma Aldrich, and they supply around 4,000 odd molecules. It's a small library, but nonetheless one can can get good hits, depending on the kind of targets that you're looking at. The beauty is they have compounds that are into different pathways. So if you are interested in looking for compound that target, say, ion channels, or say cell cycle or cytotoxic compounds, you can opt for libraries from these pathway, and then you can perform your screening. So there are other commercial vendors. So these are a list of quite a bit of them, and they can give you different diversity of compounds, and then you can use them to do your screening. Okay. So with this, we come to the next important aspect, which is the assay itself, right? So the high throughput screening requires a very powerful assay for screening your compound. So before we go into this, let me give you an idea as to how this kind of screening is done. So it's not done in a regular test tube or appendix. You're looking for screening a lot of compounds per day. So as Professor Manjunatha just mentioned, so the goal is to screen several thousands of compounds in a day. So if you just calculate, like if you're dealing with 100,000 compounds and you're screening 10,000, it will take at least like close to 10 days, right? So now if you're looking at millions of compound, it will take further time. It may be close to a month or so. So that's where people try to use different formats to screen their compounds. So it could be a 96 well plate, 384 or 1536. So these are different formats for screening their compounds. And what I would like to get your attention is that as you increase the number of wells, the reaction volume or the assay volume decreases. So if you have a 1536, yeah, you can max use a 10 microliter volume. But if you have a 96 well screening format, you are a bit okay. So you can use at least like 100 to 200 microliter or something like 2.2 ml. So this is a kind of screens, number of compounds that can be done and number of plates that can be screened per day using different formats. So you may also note that if you have a 96 well, at the maximum you can test around 80 compounds in a plate. The remaining ones will use positive control, negative controls, and so on. So you cannot use all your 96 well for testing compounds. So you can use a fraction of the 96 well. Okay, so now we'll come to the actual part of discussing about the assay. So the assays can be classified into a biochemical assay or a cell-based assay. You can also classify them into homogeneous assay, wherein it's like a mix and read. So you add some reagent to the assay and you get an output with which you can say whether it is a positive reaction or a negative reaction. So the homogeneous reactions are the most preferred of the lot for the simplicity. And of course, you also have certain heterogeneous assay, which involves multiple steps like filtration, centrifugation. For example, a common one is like ELISA, where you add antibody, you do washing. So it is much more involved. So generally people prefer the homogeneous assay because now you have the robotics involved. So a robot can be programmed to add a reagent and read rather than doing all this multi-step process. Okay, so let's look at the different examples. So this is a kind of a table chart showing different biochemical assays and cell-based assays. So you have the homogeneous, heterogeneous, and then you also have radioactive, non-radioactive. So there are different formats that are available. So we'll deal with biochemical assays first and then go to the cell-based assays. So the concept of a biochemical assay is that you have a partially purified or a completely purified protein to which you use your small molecules to screen your compounds. So in the case of COVID, I think they are using one of the non-structural protein for screening. And I think there is also, people are considering the polymerase. So essentially they are looking for one protein and against which they test different chemical molecules and look for an inhibitory molecule. And since this process requires a partially purified or a protein target, it requires cloning, expression, and purification of the desired protein. So that is a must. You cannot do without that. So, and since this is a purified assay system, it gives us a bit of flexibility. So for example, membrane permeability and serum binding and other complications with biological fluids are not a concern here. So you are just having a protein of your interest, you have a buffer where the protein works, and then you put in your compound, do an assay and see whether it works or not. Okay, so there is a bit of drawback for this. So the problem, as I mentioned, is to get this protein purified, and that is the main hurdle here. So in this kind of a chart here, it shows the success rate of cloning and purifying protein. So except for a few instances, it's a pretty poor success rate, right? So if you go to the next slide here, and I don't know if it is very clearly visible. So towards the left part of your, you have a column which has a lot of proteins. Okay, so these are all enzymes. So and you have all these bars here, black and white and so on. So the black bars indicates event when this one could do a successful screen, meaning the protein was purified, they could run a library and they got some hits, so it was a successful event. You can see from this schema in the slide that there are a lot of events that were a bit of a failure. So one has to take appropriate measures. So of course now there are some advances in the protein purification system, but still you cannot go without this purified protein because that's a requirement for biochemical screen. So now let us assume that you were successful in purifying the protein of interest, and now you are kind of trying to develop an assay for it. So what are the kind of assays that you will develop and what is the principle? So we'll look at a couple of examples. So one is the radioactive assay. It's a homogeneous assay, as I mentioned. The homogeneous assay are more preferred, so we'll deal with the homogeneous assay as such. And this is an example for ligand binding. Okay, so the principle for these sort of radioactive assay is the scintillation. So what is scintillation? So scintillation is a phenomenon when a high energy particle emits light when it is in a transparent medium. So it is called a scintillation medium. It interacts with the high energy particle, and as a result you will emit light. Okay, so in if you look at a kind of how such an assay can be designed, so let us assume that you have a protein against which you want to screen a molecule. You immobilize your protein onto a scintillant bead. Okay, so the bead is sensitive to a radioactive compound. So now once you have your protein embedded or immobilized onto this bead, then you put in your compounds of interest which are radioactively labeled. So once this compound interacts or binds to this protein of your interest, then it will transfer the energy to this molecule. As a result, it emits light. So now if there is a positive interaction, you will get light. Otherwise, you won't detect light. And this light is detected by very sensitive CCD cameras. And so you know whether there's a positive reaction or not. So this assay can be done on a 96 or a 384 platform. And usually people end up using tritium or iodine 125. These are the preferred radioactive derivatives because they are of low energy, and so there will be a scintillation event only if they are close to the molecule. So there is another assay that is a fluorescence based one. So it is a non-radioactive one. So in this study, the authors were trying to design an inhibitor for an enzyme. And since it's an enzyme, they know what is its substrate, right? So what they engineered was a fluorescent substrate, so which is called as, like, say, F here. So this is a fluorescent tag. So now if you imagine, if you have this enzyme and substrate, the enzyme will cleave this substrate. As a result, you will not detect fluorescence, or you will observe a reduction in the fluorescence. Now in this assay, if you add molecules which let us say has an inhibitor, then the enzyme activity is inhibited. As a result, you will recover fluorescence. Okay, so this is like a low fluorescence versus high fluorescence detection, and usually this can be coupled with a multiplate reader, and so you can find out whether there's a positive reaction or not. So generally the high throughput screening assays now try to employ more of the fluorescence based assays, not the radioactive ones. Mainly because first of all, whenever you deal with radioactivity, you are dealing with radioactive waste, so you have to have a proper channel of disposing it. And secondly, radioactive compounds have a shelf half-life, right? So they have a decade period. So essentially, like for if I'm not wrong, tritium is around 12 years or something, so which is relatively okay. But iodine is, I think, close to 2 months or so. So you after 60 days or close to a few months, your compound the reason people end up preferring the fluorescence based assays. So there is another non-radioactive assay, which is called alpha Lisa assay. So this is more of a proximity assay. So you're trying to find out, this assay works when two molecules are very close to each other. So let's look at what is the basis of this assay. So you have an alpha donor, which is like a red molecule, and then you have an alpha acceptor. So this is a yellow bead. So to this red bead, you attach a protein of your interest, and to the yellow bead, you attach your ligand. Okay, and then you mix them and look whether there's an interaction or not. So once there's a positive, if interaction, then these two beads are in very close proximity. So now what you do is you excite this bead with 680 nanometer. This will excite this molecule and it will transfer a singlet oxygen, which activates the acceptor bead, and it will emit light. Okay, so it's like a chemiluminescence sort of reaction, and this can be detected by CCD or other powerful methods. And so it is only activated by light and only works out when if the two molecules are in close proximity. So the close proximity is achieved only when the receptor and ligand are closely interacting. Okay, so these are some of the examples for biochemical assays. So now we look at some cell-based assays. So before we go into that, we look at what is the advantage, or why do you prefer a cell-based assay and over a biochemical assay, and what are the advantages. So the first and foremost is a cytotoxic, and compounds which do not have bioactivity are ruled out at the outset, right? So let us say you got a molecule which was working in with a purified protein, but if it is ineffective on a cell, then it is of no use. So this can be achieved, like this kind of cell-based assay will give you that power to select compounds which have bioactivity. Okay, so of course this assay is more relevant to the disease condition, because you are dealing with cells which is a biological entity rather than just an in vitro reaction. And then the other important thing is you don't require any specific protein production or scale up. So you can rule out, your success. If it is not a successful protein purification, you can always use this method for screen your compound. Of course, with advantages, you always come up with a bag of disadvantage. So one, and the most important one is, since you are using cells, you require a high volume of a pure culture, right? You don't want a contaminated culture to run your assay. So you require a high volume of culture, which is contamination free, and also you require to be maintain them in log phase or actively growing culture. So this is one requirement, and that requires quite a bit of involvement. So other foremost thing is, like, since you are dealing with cells, you have a very little flexibility in terms of the reaction condition. So in the sense, you cannot keep them at a non-physiological temperature or pH. So these are some of the problems that you have to understand before you opt for these kind of cell-based assay. And another important thing, for example, in the case of some of the infectious disease, is that you cannot do these assays in regular laboratory, because the infectious agent requires certain containment facility. So you'll probably require a BSL-3 or a P3 facility. For example, COVID requires a P3 facility to do your screening and as such any work. And so also for TB. So you cannot do it in regular lab. So these are some of the disadvantages. Of course, the pharma industry definitely has all these disadvantages taken care of. So now let's look into some important or famous cell-based assays. So one of the most widely used is a reporter gene-based assay. So here what you use is called a reporter gene. So the reporter gene could be a fluorescent protein or a bioluminescent protein like luciferase or so on. So such a protein is tagged onto a conditional promoter. Okay, so this is the concept behind reporter gene. So you have a conditional promoter which is fused to a reporter gene, and then this entire setup is in a cell, or it could be a mammalian cell or a bacterial cell, whatever. So now the idea is you will put in your compounds of interest. So this is your extracellular stimuli. So the compound is expected to stimulate a signaling pathway which will activate this promoter. So once you add a molecule, it will activate the promoter, and once the promoter is active, you will get the expression of your gene, which can be read out by a fluorescence or a luminometer reading. So the other widely used is the dye reduction based assay. So this is very commonly used for antibiotics and cytotoxic screens. So some of the students in our lab use this assay for screening antimicrobial activity compounds. So this uses a dye called risazurin, and it's a dye reduction based assay. So what is the principle here? You have a dye which is having a very low fluorescence, and now when you add it to a culture, because the cell is actively multiplying and it is having an active metabolism, it will reduce this compound to a highly fluorescent compound. Okay, so now you add this in your assay and then you put in your test compounds. So if the compound kills a bacteria or a cell, then the metabolism is affected, and the result you will see like blue or purple colored spots or wells. So this indicates the molecule is cytotoxic or probably antibacterial. And if the molecule is inactive, then the cells grow and metabolizes, and as a result, the dye gets reduced and you see them in pink color. Okay. Okay, so now let us go back and assume that you started out with a protein, and then you got established an assay, and now you want to find out how this assay works, like whether it's a good enough an assay to identify the compound. So if your assay is not good, then your rate or your idea of drug discovery is completely in soup. So here what is called here is what is called the Z factor for assessing the robustness of your assay. Okay, let's look at what is the Z factor. So the Z factor essentially tells how well are the test sample and your control samples are separated. So whenever you do a fluorescence assay, so you get a fluorescent reading and you'll have a negative control which will not give you a fluorescent reading. So how well these are distinguishable. So that is the whole idea of this Z factor, and it is calculated using this formula that is listed here. And you look for Z scores that usually lie between 1 to 0.5. Okay, so if you get 1, that's a very ideal assay, the best assay that you can get, but usually under laboratory condition, it's quite difficult to get an ideal assay. So you look for Z factor anywhere between 1 to 0.5. So, okay. So now let's look at how this translates. So this is an example of a high throughput assay which is standardized, and now they are trying to evaluate the robustness of this assay. So you have an example A and an example B. So let's look at the example A. So you can see that there are compounds, like there's a good separation between the control sample with the test sample. So these are your positive or the assays that give you a readout, and these are the negative controls. So there's a very good separation between these two, and you also see that the Z factor is around 0.5. So now you look at assay B, you see that the outcome is scattered all over the place. And in fact, it is very close to the negative control, right? So as a result, the Z factor is 0.1. So this is not a very good assay. And if you have a chance, you definitely would prefer an assay which has a Z score of 0.5 or above, and not such assays. Of course, once you have done this kind of a Z-score analysis, we'll also look for reproducibility, in the sense you will do an assay with, say, initially before going for a 100,000 compound, you'll probably run a assay with, say, around 10,000 compound and test what you get the outcome, and probably repeat it after 1 or 2 days to see if the outcomes are reproducible. So these are kind of pilot screens, what we call, before you run a full-fledged high throughput screen. So you know your assay is fine, works beautifully well, it is reproducible. And so once you have made sure all of this, you run your high throughput screen. So when you run your high throughput screen, you end up screening like hundreds and thousands of compounds. And now this eventually generates tons of data, right? So you are getting, like, if you are taking 100,000 compounds, you are getting 100,000 points, and you you need a kind of very good informatic backup to analyze and get some sense out of this. So there are different companies which sell you analytical packages. Of course, one has to buy, and industries or even academic institutions which run high throughput screen have all of this inbuilt. So it all comes as a package. So now your high throughput screen is running and you get all sorts of data, and so you can plot the graphs, you know whether which compound is working. So everything is well laid out. Okay. So now there is something called a virtual screen. So what is this? So in some of the introduction slides, I was mentioning that we have a huge chemical space, somewhere around 10 raised to 60 or so. Okay, so now you have a huge number of chemical molecules. Many of them can be, a small part of them are synthesized, but you can always generate them in silico. So in a computer, you can design so many of these compounds. And now with the advances in our structure determination, we also have a lot of protein structures that are available. Of course, you can also have very powerful algorithms which can model proteins as well, right? So if you submit a protein sequence, a primary amino acid sequence, they can model to give an idea about what a final protein structure would look like. So with these advances in the structure and availability of a lot of chemical space and further advances in our computation, one can think of doing all of this interaction in silico. So in the sense, you take your protein, you take some molecule and see whether they interact or not in your computer itself. So you can do that sitting at home. So what is the advantage? You can test a lot of compounds and then find out what are the compounds that are potentially binding, and then you can synthesize only the compounds of interest. So you don't have to synthesize millions of compounds. You can select a few of compounds and then test them in experiment, right? So so this is what I meant. Because of the advances in our structure, like you have X-ray, you have cryo-EM, NMR. So due to the advances in structure determination, we have a lot of protein structures that are currently available. And now you can also use several algorithms and softwares to do your docking studies. So some of them are laid out here. You have Monte Carlo, you have Autodock. Some of them are, in fact, I think Autodock is freely available. So you can just download this program. If you have a protein and you have access to chemical library, then you perform docking, and then you'll get a docking score based on which you can know whether there's a good interaction or not. Of course, there are some advanced softwares like Schrodinger, which can do a lot more. Like they can find out whether the molecule is interacting, and they can also suggest what are the potential derivatives which will have a better activity. So you have a lot more flexibility with Schrodinger. Okay, so if you are closely following the HRD ministry, I think now it is the education ministry. So within the government of India is running a drug discovery hackathon program. So the main challenge or the goal of this hackathon is to identify molecules against the COVID-19 by an in silico drug discovery approach. Okay, so now the idea is anyone can get involved in this and take part in the drug discovery program. Of course, you have some of the partners, Schrodinger and Chemaxon. These are the your software partners who are also involved in this process. And you can see there are different problem statements. So essentially, it is like you are asked to test chemical compounds, whether they bind to the protein. I think it could be one of the non-structural protein, or I haven't, yeah, I think it is one of the non-structural proteins that is used for this high throughputs in the in silico screening. And once you come up with deposit a molecule of interest, then they'll try to do lead optimization and look for other ADMET scores and so on. Okay. Okay, so now we have come this far. So we started out with library of compounds, understood how they are synthesized. Then we looked at some assays, biochemical and cell-based assays, how different assays are used. And then you also got an idea about what is virtual screening. So now let us assume you could do an assay and you got a molecule. Okay, so now what is the target of the molecule. So if you are using a biochemical assay, by definition, you know what is the protein that you are using. So there the target is already known. So you don't have to worry too much about understanding the target. But if you're using a cell-based assay or like a cytotoxic or phenotypic based assay, like you add a molecule and it causes um, um, bacterial death, right? So you don't know what pathway is being targeted, how the bacteria is dying. So you want to understand what is a target. So in order to do that, there are certain approaches that are used. So we'll look a couple of them. So you can employ a proteomics based approach or a genome based approach. And once you have identified the target, you can further validate by genetic methods. Say you can use CRISPR or siRNA based methods. So essentially find out whether the target is actually hitting the gene that you have identified from your target screen. Okay. So now let us look at a proteomic based approach. So this is from Schreiber's group, who we talked in the introduction. So he's one of the pioneers of the chemical biology field. And to give you a brief idea about this study, so the study was designed to understand or identify components of rapamycin signaling. Okay, so this is called a TOR signaling. It's a very important signaling pathway, which is conserved from yeast to humans. Okay, it's a very important signaling pathway which governs several cell process proliferation, migration, cell division, and whatnot. So the idea was to find out other components that act in the signaling network. So this is the way the assay was designed. So you have yeast cells. So all this was done in yeast cells. So you take yeast and you add your compound library. Okay, so they have used around 16,000 odd compounds, which is relatively a good number of compounds to test. And in addition, they are their aim was to identify whichever work in association with rapamycin. So rapamycin is also a part of this assay. So now let's look at the readout that they are looking for. So now if you use rapamycin will inhibit the TOR signaling, and as a result, it limits the cell proliferation. Okay, so now if you add rapamycin and you incubate for days, you get a little bit proliferation. Okay, so now in this library, if you have a compound which actually inhibits rapamycin, okay, so if it inhibits rapamycin, then it can no longer inhibit the signaling, TOR signaling, and therefore you will get very good proliferation of cell. So this is depicted here. You have a lot of growth, and these are kind of classified as rapamycin inhibitor, SMIRs. Okay, and now you can have another outcome wherein it enhances the activity of rapamycin. So it will become, say, rapamycin plus plus. So as a result, the growth is much reduced compared to rapamycin alone. So these are called the enhancers of rapamycin. Okay, so in this assay, they performed this kind of a screening assay, and they got like inhibitors of rapamycin, enhancers of rapamycin. So these are some of the chemical structures they have put across here. And now the idea is to find out what is the actual protein that these molecules are hitting. Okay, so since this is a yeast, they made a yeast proteome array. So in the sense, they spotted each protein of the yeast onto a imageable platform, could be a glass slide, and then all these proteins are kind of spotted and they are immobilized. So now you just flood this slide with the molecule. Okay, so for example, you want to identify the inhibitor 1, SMIR1, you just flood this molecule onto this array slide. And this molecule is conjugated with biotin. Okay, so this is important here. So now once you allow this molecule to bind to its target, then you flush out the unbound molecule, and then you probe the molecule by streptavidin. For people who are not aware, that streptavidin and biotin form a tightly interacting partners. They are very specific, and it's very specific and very tight. So once you bind, it is almost like irreversible. You can very stably find an interaction. So now what they do is they attach a fluorescent molecules to streptavidin. So wherever streptavidin binds, you can image that spot by a fluorimeter or a fluorescence reader. Okay, so this is what exactly was done. So they flood with biotin conjugated molecule, they wash it, then put streptavidin conjugated, fluorescent conjugated streptavidin molecule, and once they imaged, they got few spots. Okay, and since this you already know which spot corresponds to what protein, you can go back to your notebook and check out what this protein is. So there are some proteins that are listed out here, and so this way you can identify the target. So this is totally proteome based approach. Okay, so now we look at another example wherein a genomic approach is being used. So before we go, I'll just give you an idea about what is the approach that was used. So in this study, the authors used a forward genetics approach. So what is forward genetics? You have a phenotype, and then, so for example, you have, say for example, cytotoxicity is a phenotype, or bacterial death is a phenotype. So now once you have a phenotype, you look for what is the gene that is causing this phenotype. Okay, so this can be done by several genetic methods, which are listed here, and we'll look into the example to understand it in bit detail. Okay, so this was a paper wherein the authors were interested in finding an anti-tuberculosis molecule that affects mycolic acid. So for people who are not aware of mycobacteria, so mycobacteria has a complex cell wall structure. So in addition to peptidoglycan, it has additional molecules called the mycolic acid, arabinogalactans, and this complexity of the cell wall, what makes it very resistant to several antibiotics. So it's like a shield, essentially like a shield that gives an inbuilt protection to several antibiotics and so on. So the concept here is if you can affect or destroy the mycolic acid, you can affect bacterial viability, make them sensitive to antibiotics, and in fact, you can kill them. So in fact, one of the frontline drugs, isoniazid, affects mycolic acid, and so it's a routinely used antibiotic for TB therapy. So coming back to the paper, so in this study, a group had already screened around 100,000 compounds, and they had reported out of this 100,000 compound, around 1,000 or so had anti-tuberculosis activity. So now these authors were interested to find out if any of these 1,000 compounds had could affect mycolic acid biosynthesis. So that's what was their interest. And to understand, to identify such compound, they used a reporter-based assay. Okay, so now let's look at what is a reporter. We already saw this in our cell-based assay. So here they used a promoter called INIBAC. Okay, so the INIBAC is a conditional promoter, and a LAGZ reporter, which is a kind of chromogenic reporter, is attached to INIBAC. So what is the principle of this assay? So when a molecule affects the cell wall, or it hits the mycolic acid, then it will initiate a signaling pathway which will lead to activation of INIBAC. So if there is an INIBAC activation, it means there is a cell wall damage. Okay, so now since you have the compound, you just throw in your compounds and look which wells give you LAGZ activity. So that is the idea behind this. So eventually, when they did this screen, they got a good molecule, and that also showed antimycobacterial activity. In the sense, if you use that molecule, the bacteria is dead. So it is essentially working like an antibiotic. So now they want to understand. So it's like a phenotype, antibiotic-based, molecule-based bacterial death. So they want to understand what is the mechanism that causes, or what is the protein that this molecule is targeting. Okay, so to understand this, they employed a forward genetics approach. So the concept here is the molecule causes bacterial death. Okay, so now if you use you can if you grow a good population of bacteria, there could be one or two mutants which kind of develop resistance, which are having any resistance to this molecule. So what they did in this experiment, they grow the bacterial culture, plated on agar medium that contains this antibiotic, and then try to obtain resistant mutants. Okay, so they got the resistant mutants, and then what they did was to isolate the genomic DNA and did whole genome sequencing. So in the current age, whole genome sequencing is affordable, and one can get pretty good data of the entire genome. Okay, so now they have sequenced the genome of the mutant, which is resistant to this antibiotic. So now when they analyze the genome sequence, they could find a mutation in the PKS13 gene. So this was phenylalanine to serine substitution, and it occurred in this PKS13. So PKS is a polyketide synthesis enzyme. So it is involved in modifying the lipids, which eventually get incorporated into the cell wall, part of the mycolic acid chain. So then to further validate whether this is actual target or not, they purified this protein, then tested whether if you add the molecule to this protein, if it affects the conversion, whether if it affects the enzyme activity. So these were all the other background experiments that were done to validate. Okay, so this just gives you a flavor how modern biology methods can be used to identify a target even without knowing what is like based on a cell-based screen. Okay, so with this we come to the summary. So let us look at the journey of chemotherapy. It's been quite exciting, and it's almost like more than 100 years now. So it all began with Ehrlich, who gave us the concept of magic bullet and initiated the field of chemotherapy. And this was followed by Fleming's discovery of antibiotics, which unleashed an era of natural products. Okay, there was a flurry of antibiotics screened from 1940s to 80s, and they still continue to be discovered. And then came Schreiber and his colleagues, who developed or were the pioneers of chemical biology. Then brought this interdisciplinary working of chemistry and biology to identify drugs. And then using the concepts of chemical biology, the pharma industry jumped into the picture. They brought in their automation into this, allowing to screen thousands of compounds in a go and identify interesting drug-like molecules. And now with the advances in our computation and structural determination, we can do most of our screening in silico. And based on this in silico screening, one can identify lead molecules and then go back to your chemistry lab, synthesize selective compounds, and then test whether they work in vivo or not. So essentially, you're trying to cut down the time of identifying your lead molecule. Okay, so now with the advent of artificial intelligence, many companies and many academic institutions are also looking whether artificial intelligence can be employed in drug discovery. Okay, so this is the future. It's kind of an upcoming field, and looks exciting. We are and hope sooner we can come up with a drug to the pandemic and everyone is back to life normally. Okay, with this, I thank you all for your attention, and I'll be happy to take questions. Okay, I can't.
Thank you, sir, for the enlightening us with the splendid talk on drug discovery. Just to show you, know that we have got a lot of appreciation for the
thanks.
talk. It was very understandable.
Yeah.
So now the session is open for questions. I kindly request all the participants to drop the questions in the chat box.
Yeah, feel free, because I can even send some reading material if you're interested, because I just kept the material general. So I did not involve too much of complexities here. So if you want, we can send you selective reading material. If someone is interested, you are most welcome to contact me.
So here we have one question from Varsha Shetty. Okay. What is the material of bead in alpha Lisa technique?
Okay, so these are usually inert molecules. So usually these this material is, I'm not sure if they are kind of disclosed. So these are inert molecules which have some linkers. So which will allow you to conjugate your protein of interest. Okay, I'm not very much sure if I can find it out if what are the available composition. Usually it will be some sort of matrix which is amenable to certain chemistries. Could be some polysaccharides and so on. But exact molecule can vary because you want to know which protein you want to conjugate. So it varies from protein to protein. So you'll have a plethora of chemistries. So yeah.
Here is one more question from Vaishali Rai. How many assays has to be done to prove the activity of the compound?
You mean high throughput assay? Or I didn't get the question. How many
How
assays need
many
to be done?
assays has to be done to prove the activity of a compound?
Okay, so usually if it is an enzyme based assay, right, so you the way usually these high throughput screens are done, you initially start with one concentration. Okay, so the high throughput screening is the idea to screen thousands of compound to get a lead molecule. So once they get a lead molecule, they'll probably look what is the concentration. So you'll probably start with a blanket concentration of 10 micromolar or something, and then once you get a lead molecule, you identify whether the concentration is effective or not. But there's no concept like how many times you do because your assay is inbuilt. You have made sure that your assay works very well. So there's no idea of going back to repeating that assay. What you will probably end up doing is to find an effective concentration which can inhibit your enzyme, because that is what is our interest. So if the enzyme is inhibited at 10 micromolar, can it be inactivated at say 1 micromolar or 0.1 micromolar? So that will be our interest. Of course, people do it probably 2 to 3 times to check reproducibility. Because if the molecule if it was a fluke event that it worked once, so they repeat it once more with different concentrations. So that is kind of a reproducibility test.
There's one more. What is TRE under the reporter gene topic?
Oh, it is transcript, like it's a enhancer element transcription. So it's like the example was a mammalian system. So where you have enhancer, you have transcript. So you have promoter, enhancer elements. So it is just a schematic. Essentially, you want a promoter, regulatable unit, which will govern the expression of a reporter gene. So it usually will be a promoter with all the components that will allow the expression of gene in a conditional fashion.
Okay, there is one more program for evaluating docking results.
So this like Autodoc is one program. I think Autodoc is, if I understand, is freely available. So I need to look into what are the actual docking scores. Like, so essentially you take a molecule and you need to convert into a format. So this is all a computational method. So you will take your PDB, you take your molecule, and then you convert them into a format which the system reads. So then you perform a docking, and then it will give docking scores. So based on the docking score, you understand whether it's a good interaction or a weak interaction. So based on this docking scores, you can evaluate whether a compound A is showing binding activity. The docking will not tell whether it's an inhibitor or not. Okay, so it will just tell you whether there's a good interaction or not. You may have to probably, like if you know the structure, you may understand where it is interacting, so you can find out whether it is in fact inhibiting the activity or not. Of course, once you do all your in silico experiments, so this is one advantage and probably kind of a drawback of the in silico system. The in silico system allows you to do at home. Okay, so essentially the drug hackathon is like you can do it at your home. You just enroll into that program. You are probably even given the software, so you can test it at home. But now once you have got an inhibitor or an interacting molecule, that needs to be evaluated whether it is functionally active or not. So for that, you kind of go back to your lab, do an experiment, put that molecule, check whether it affects the viral replication and whatnot. So it requires a wet lab experiment confirmation. But in silico, like you can leverage upon it. So we cannot do at this time, go to a BSL-3 lab and do a COVID screening. You are most welcome to look into the molecular space and identify some compound. In fact, that's the competition all about. Yeah.
Vinay D'Souza have asked a question. What is the importance of silicon study before conducting an in vivo study on the effect of antagonistic receptor interaction?
Oh. Okay, so as I was just talking right now, the in silico many a times can be done in parallel. Okay, so if you have access to the molecules, the most of these molecules are available in repositories, so they are available online. So essentially all this in silico method convert your molecule into some kind of a vector. Okay, I think that's what they are called. So you convert a double bond into some parameter which the computer program can understand. So you can use this to do your docking study, and once you get a hit, you can experiment and validate in vivo. So that way you can reduce the number of compounds that you synthesize. uh, did that answer the question? I don't know. So the idea is you can instead of testing thousands of compounds, you can narrow down onto selective compounds. So instead of making 10,000 compound by docking studies, by in silico approaches, you can probably narrow down upon to say something like 10 or 50 compounds. And now you synthesize them, find out whether they are bioactive. And once you get bioactivity, then you again have to make some derivatives of them to optimize. Some of them can give better activity, less activity, and so on. So essentially this in silico reduces your what to say,